Hey everyone, this is Mr. Baker. We are going over our fourth section of Unit 3. We're going to talk about how DNA is used to make proteins. What are we doing with all that genetic info that's being stored on the DNA? Remember that proteins from 2.5 were what we use to accomplish jobs and give traits to organisms. And we know that DNA is our genetic code. So this is the mechanics of how that works. We aren't going to go too far in depth here, but I want you to know that this process is sometimes called the central dogma because it is so important to biology. Right? It is this process of going from DNA to an RNA sequence to a protein itself. The first step from DNA to RNA is called transcription. Transcription, I always remember that it comes first because alphabetically it'll come first between transcription and translation, right? This works exactly like DNA replication, except for mRNA, we're making a single strand and there's no thymine in it. So when we go to do our matchups, as you see on screen, adenine does not pair with thymine. It pairs with another nucleic acid called uracil. Right? So we've got a slightly different nucleotide there. It works the same way. Right? I have had students forget about thymine, but after we do this a couple of times, you'll get the hang of mRNA not having any thymine in it. Transcription is important because DNA is stuck inside the nucleus and it's got all the information in it. So to get that information out, we have to make it into mRNA. We are copying the DNA template strand into an mRNA. All right. The DNA will stay in the nucleus. The mRNA can be brought out. So just as a quick way to apply that, I have done some transcription here, but I made some mistakes. And I want you to circle the mistakes on the mRNA strand that was created based on the template strand. So write these down, circle the errors, and I'll check those as part of grading your notes. Before mRNA can be used, it needs to be processed. mRNA processing is a way that cells are able to only use the part of the DNA that actually codes for the protein or the polypeptide. So it's going to remove introns. It's also going to protect the mRNA strand by giving it a cap. And then it will remove the 3' end that's extra because when we go through transcription, it actually processes it. I don't want to use the word process. It transcribes more DNA than is necessary, right? So we've got to remove the 3' tail and add what we call a poly A tail. It's a long string of adenine. And that works to protect the mRNA as it travels through and as it's used. Lastly, mRNA processing goes through something called splicing, where introns are removed. Introns are extra information in the DNA that are used to turn on or turn off transcription as a way to control whether that gene is being expressed or not. We'll talk more about that later. For translation, our second part, we are going to take that mRNA and at a ribosome or a rough endoplasmic reticulum that has ribosomes on it, it's going to feed that mRNA through the ribosome, right? That's called initiation. The ribosome is going to connect amino acids that are brought to it from tRNAs in a specific order dictated by that mRNA to form a chain. Once the string of amino acids, which we call a polypeptide, is done being assembled, it's released from the ribosome and it is processed, usually through a process called protein folding, because proteins not only need the right set of amino acids, they also need to be the right shape in order to do their job. So protein folding finishes that up. We are not going to talk about protein folding very much. We're focusing on initiation, elongation, and that release and termination. So on a ribosome, we've got two parts, the large ribosomal subunit and the small ribosomal subunit. they are going to pinch around the mRNA and travel along it in a 5' to 3' direction, right? So the A on the right is the front. I like to think of that as arrival A, right? A tRNA is going to come in and its anticodon, its set of 3 nucleotides, are going to match up to the codon found on the mRNA. Once that happens, the ribosome is going to shift down a set of codons, and the anticodon and its accompanying amino acid are going to go to the P site for processing. That's where the ribosome is going to connect amino acids together. And E is for exit. It's where the amino acid is actually broken off of the tRNA, and the tRNA is released back to the cytoplasm. Here's another view of that kind of working as a cycle. And one that I like better is actually this diagram, where we can see the mRNA moving. Again, it is moving towards the 3' end. So A is first, P is second, E is last. Arrival, process, exit, or empty. In our class, we aren't going to look at tRNA and figure out their shapes. Instead, we're going to use the codons on the mRNA and this diagram. This is a mRNA codon chart, right? It's going to show us which amino acids are connected to which mRNA codons. So it kind of works and lets us decipher mRNA. So if we're going to use this, let's walk through it pretty quickly, right? Here is our codon sequence in the top left. What we're going to do is we're going to start on the left side, and we're going to look at that first letter being A. So I'm just blocking off the ones we don't need. Right? Our second letter is U, so we know that it's in the first column. And then our last letter is G. So inside that cell, G is the last one. Methionine is MET for short. We're going to keep using those 3-letter abbreviations. And it is called a start codon because it is very, very frequently found at the start of polypeptide chains, but it can also appear in the middle and towards the end, but it won't be the very last, and it is almost always the start. As we practice this in class, sometimes I will start you out with this, but I will not always start you out with this. So the second one is AGU, so we're still in that third row, but now we need to go to the far right-hand side as our second letter is G. Our last letter is U, so that's serine, S-E-R, right? We're almost done. The third letter is U, so that tells us we're in the top row. Our second letter is G, so that's the far right-hand column. And then we look at the third letter being A, and it gives us stop instead of a 3-letter abbreviation. Stop means stop. We are done transcribing a polypeptide sequence, and it's going to tell the ribosome the exact same thing. We are done. You can release the polypeptide chain, and you can release the mRNA. For us, we are going to write the word stop every time we get to a stop codon so that we can, both I and you, understand that's what that means, because I need to make sure that you understand. This is another version that I will give you in class. I will give you the first one we had and this one on the back of a piece of paper. It works the exact same way. It still uses mRNA codons, but instead it starts in the center at that 5' part and works its way out towards the 3', which is on the outside. We will practice transcription and translation in class as we continue to talk about how the code of DNA is used and how it makes proteins in cells. Make sure you've got everything in your notes. Go back and watch this again if you need to. Write down any questions you have. I will see you in class.